Diffusion and osmosis. The function of a cell's membrane is to provide a barrier for intracellular materials and to serve as a gateway allowing materials to enter and exit the cell. Membranes which allow some molecules to pass and reject the passage of others is called semi-permeable. The transportation of molecules requiring energy is called active transport. The transportation of molecules without the use of energy is called passive transport. Diffusion is the movement of molecules from higher areas of concentration to lower areas of concentration. Osmosis is the diffusion of water across a semi-permeable membrane. This experiment is designed to model diffusion across a membrane through the use of dialysis tubing. After 30 minutes went by, the dialysis bag turned from an initial clear color to a navy blue color. The glucose test strips also revealed evidence of diffusion. The solution in the cup initially tested negative for glucose, but after 30 minutes it tested positive. Likewise, the dialysis bag initially tested positive for glucose, but tested negative after 30 minutes went by. This indicates that the glucose diffused from the high concentration to a low concentration. Similarly, the change in color and increase in size of the dialysis tubing proves the diffusion of iodine into the dialysis bag. However, because of the size of the starch molecules, they were not able to diffuse across the dialysis bag into the IKI solution. This experiment is designed to investigate the influence of solute concentration on the net movement of water molecules across a semi-permeable membrane. Our results vary depending on the molar concentration of sucrose present in the cup. The process of osmosis was indicated through the change in mass of the dialysis bags before and after being immersed in the sucrose solutions. Since the final mass was more than the initial mass of the dialysis bags, it symbolizes the diffusion of a solution across the dialysis tubing. The higher concentration of the sucrose became, the higher the net movement across the dialysis bag. The movement of water in and out of a cell is shaped by the concentration of solute on either side of the membrane. Water potential is a concept that uses a combination of the difference in solute concentration and pressure to predict the direction in which water will diffuse through plant tissue. Solute potential, which is dependent on solute concentration, and pressure potential, which results from an exertion of pressure, are the two major components of water potential. The net movement of water is always from an area of high water potential to an area of low water potential. This experiment is designed to investigate water potential by submerging potato cores into different sucrose solutions and comparing their changes in mass. In conclusion, the concentration of sucrose solution greatly affected the changes in mass of the potato section. The final mass of the potato cores was less than the initial mass. The higher the molarity of the sucrose solution, the more the mass of the potatoes decreased. The net movement of water in this experiment went outside the solute, the potato, and into the sucrose solution, due to the fact that the water potential was higher in the potato cores than in the sucrose solution.